Have you ever wondered how a normal, healthy cell becomes cancer? It may not work the way you think it does. It isn't one bad mutation flipping a switch just like that. Every cancer begins the same way, as one normal cell that goes wrong, then keeps going wrong. This is a healthy cell. Every day it divides, copies its DNA, and follows the rules. Certain things damage that DNA. Scientists call them carcinogens or mutagens. Sunlight's UV rays, tobacco smoke, even random copying mistakes made every time a cell divides. It happens more often than you'd think. A single cell can rack up tens of thousands of these damaging events in a single day. Most of the time, the damage gets repaired or the cell dies. But once in a while, a mutation slips through, especially in a gene that controls growth. That cell is now initiated, predisposed to cancer, though not cancerous yet. To actually become cancer, that initiated cell needs more than one mutation. Over time, as it divides, it picks up additional changes, each one unlocking a specific ability researchers call a hallmark of cancer. One mutation hijacks its own growth switches. Genes called oncogenes get stuck in the on position, telling the cell to divide nonstop. Another disables its safety brakes. Genes like P53 normally catch damaged cells and stop them. But in cancer, those tumor suppressors get mutated or switched off. Often, that same P53 loss also cripples the cell's self-destruct signal, a process called apoptosis, so it survives when a healthy cell would have died. One more hallmark. Immune cells normally patrol for abnormal cells and destroy them. This one mutates further, masking the markers that would flag it as a threat, so the immune system passes it by. Now it's a transformed cell, one that's broken the rules for good. It starts to multiply fast. One cell becomes many in a process called clonal expansion. The tissue starts to look abnormal under a microscope, a stage called dysplasia, often known outside the lab as precancerous. As the cells keep dividing, they pick up even more mutations, splitting into competing subpopulations called subclones, each one a slightly different version of the same tumor. Eventually, they form a small, self-contained tumor, a mass of genetically altered cells still confined to where it started. But a tumor can't grow much larger without its own blood supply. So it sends out a signal called VEGF, short for vascular endothelial growth factor, telling the body to build new blood vessels straight into it. That process is called angiogenesis. With its own bloodline, the tumor grows faster than ever. Some cells break away entirely, invading nearby tissue and slipping into a blood or lymph vessel, a step called intravasation. Carried through the bloodstream, they can lodge in distant organs, the liver, the lungs, and start the whole process over again as secondary tumors. This is metastasis, cancer spreading through the body. What looks like one disease is really a long chain of small, specific changes, from one normal cell to a tumor that's spread through the body. And it's a chain that touches nearly all of us. The exact numbers vary depending on where you live, but approximately 2 in 5 people will face some form of cancer in their lifetime. Every one of those steps, though, is a potential point of intervention, a place where research can look and treatment can act. That's why this matters. The better we understand how cancer happens step by step, the better we get at stopping it.